Assalamu alaikum, everyone. My name is Raghda Adwan. I'm a third-year medical student. I hope everyone's Ramadan is going well. I'm sure most of the people watching this right now are probably watching it around final season, cramming for their MOL final. So my goal with this PAL, inshallah, is to make this lecture as easily digestible and easy to get through and quick as possible. Now, one thing is that this lecture is generally, it's pretty easy. So I don't want to, extensively get into any details that are unnecessary. Everything in the slides went straight from your slides, and I took note of anything mentioned in the transcription, what to focus on, what not to focus on, things like that. But just to clarify that I didn't add much that is additional. I am just sticking to what you need to know. So inshallah, this should be easy and simple and straightforward. All right, let's get into it. All right. So this lecture is divided into two halves. I decided to divide it into one half about everything about cholesterol, and then the second half all about steroid hormones. So we'll begin with cholesterol. What is it? Where does it come from? Cholesterol versus cholesterol esters, the synthesis, regulation, bile acid, bile salt, and there are two clinical conditions that you need to keep in mind: hypercholesterolemia and gallstones, which I will get into. And then the second half about steroid hormones, what they are, how they're synthesized, and then finally congenital adrenal hyperplasia. So let's begin with cholesterol. Cholesterol, I'm sure most of you think of cholesterol as this very bad thing that comes from like really unhealthy food. That's kind of the reputation that cholesterol has. However, cholesterol is actually a really important molecule in our bodies, and we actually make a lot of cholesterol ourselves rather than from consuming it in food. So cholesterol itself is a lipid. It's a type of lipid just like triglycerides. It's a lipid in the form of a steroid alcohol or sterol, and it's just a different form of lipids. So we need to sort of brief a bit about the structure of cholesterol itself. So you obviously don't need to know exactly every part of the structure, but it's important to be a little bit familiar with it. So it's called a sterol because it has this steroid nucleus, which are four fused rings, and this is what makes it a steroid. Now, what makes it a steroid alcohol or a sterol is this OH group or hydroxyl group, which is what makes it an alcohol. That's why it's called a sterol. And at the other end, there is a hydrocarbon side chain. So if you total all these carbons up, there will be 27 carbons in total. Again, you don't need to memorize the structure, but it's important because it's relevant when it comes to transport and synthesis and so on and so forth. So some properties about cholesterol: it has very low solubility in water. Now, it exists in two forms: free cholesterol and cholesterol ester. So cholesterol ester is just an esterified form of the free cholesterol. And what that means is when you have this bond right here, COO bond, that's the ester bond, if you guys remember anything from high school chemistry. That COO bond is formed when you have the free cholesterol OH group and it reacts with a fatty acid, acyl or fatty acid group. And this forms the ester bond, making it a cholesterol ester. Now, this changes its properties a little bit. That's why this is relevant, because it becomes much less soluble. So it goes from being pretty low, having low solubility, to being even less soluble as a cholesterol ester. And this is the form in which the majority of cholesterol exists in our bodies, as cholesterol ester. Now, why are we making it from low solubility to even lower solubility? I'll explain in a minute. Now, what are the functions of cholesterol? What it does in our bodies? It has multiple functions, one of which being that it's in all of the cell membranes. It's there for fluidity, for forming the structure of the lipid bilayer. The second thing it does is it acts as a precursor. It's a base molecule to make a bunch of other molecules, of which being bile, which we will have a whole section to discuss bile and bile acids and bile salts, as well as vitamin D and steroid hormones, which is the second half of this lecture. Now, before we get into cholesterol esters and transport and all of those things, we need to touch base on lipoproteins. So you guys have actually two whole lectures about lipoproteins, everything about them, and those will be discussed thoroughly. However, it would be remiss if I didn't talk about lipoproteins before I explain to you the transport of cholesterol, because that is very relevant here. And I'm pretty sure you guys don't know much about lipoproteins before those lectures. So let me just explain briefly. A lipoprotein is a molecule which is a mixture of lipids and proteins. From the name, lipids and proteins together. Now, what's the purpose of them? Their purpose is to transport hydrophobic lipids. when you have something that's hydrophobic, that has very low solubility, we cannot transport it in the blood alone. It needs to be complexed into something or put into some kind of molecule in order to help it transport in the blood, because the blood is, most of it is it's a watery plasma. You need something that will bode well with the water in the plasma. And so what we bring in to help transport these lipids and cholesterol and etc. are lipoproteins. So this is the reason why we transform cholesterol into cholesterol esters, to make them even less soluble. Now, this kind of sounds counterintuitive, because it's like, why are we making something less soluble to transport it if we need it to become soluble in order to transport it? It's because of the nature of these lipoproteins. So the lipoproteins are spherical in shape. They have a core and an outer shell. So what matters is that the outer shell is hydrophilic, and it can, it's relatively soluble, or it can form bonds with the water in the plasma. But the core is hydrophobic. It can be as hydrophobic as it needs to be. So we put all of the cholesterol esters in this core in the lipoprotein, and that allows it to be transported efficiently. So let me just mention them quickly, because I will mention them later on in the lecture. The biggest form is a chylomicron, and if it gets a little bit smaller, it's called a chylomicron remnant. Then we have VLDLs, or very low density lipoproteins, IDLs, intermediate density lipoproteins, and LDLs, or low density lipoproteins. Now, the good form of lipoprotein is HDL, or high density lipoprotein. All right, so this is relevant later on, and that is why I'm mentioning here, but you have 2 full lectures about it, so don't worry about it too much for now. All right, cholesterol esters, the esterified form. Again, this is just for the sake of repetition, to really hammer in the idea that we're transforming cholesterol into something less soluble by making an ester bond, by reacting it with a fatty acid, in order to more efficiently transport it. Now, how are we more efficiently transporting it? Okay, I talked about lipoproteins, I talked about the hydrophobic core, but why are we making it hydrophobic and putting it in the core? Why are we just not making it more soluble? So this concept, this is why I mentioned the lipoproteins earlier, because we have, for example, HDLs. HDLs is one form of lipoprotein, and it is one of the transport molecules that is carrying cholesterol. And so the immature form of an HDL has a sort of discoidal shape. It's not a full sphere. So in order for it to become a mature sphere and more efficiently move in the blood, it needs to be in the form of a sphere and not in a form of a disc. And how this happens is by packing it in with hydrophobic lipids in the center, because when we have things that are more hydrophobic in the center, they will push away the things that are hydrophilic on the outside, and therefore they will naturally form a spherical shape. And this is, by the laws of nature, the most efficient way of transport is in the form of a sphere and not in the form of a disc. This sentence here was in your lecture and it was not explained, and so I thought it was important for me to illustrate this by using this diagram, but this diagram wasn't in your lecture, the explanation of it was. So this is just to understand why we're making cholesterol esters to begin with, in order to make HDLs spherical and more mature, so that they can be efficiently transported in the plasma. Now, let's get into the sources of cholesterol, how we make cholesterol, and all of that good stuff. So we're going to talk about this in relation to the liver. We call this hepatic cholesterol. Why the liver? The liver is a major site of metabolism, of synthesis of cholesterol, and that's why we are mostly talking in the sense of liver cholesterol or hepatic cholesterol. So we have sources, and then what we do with the cholesterol that enters liver. So let's begin with the source of cholesterol. Now, let me skip that slide and go back to it. But the major source of our cholesterol comes from de novo synthesis. De novo means just synthesis from base molecules, like smaller molecules, building up something big from something smaller, rather than breaking something down. That is a different type of synthesis. So the major form of cholesterol that we make is through de novo synthesis. And we have other sources. Of course, one being the diet. So you eat food, you have cholesterol in food. Healthy cholesterol comes from things like eggs, animal products, but of course there's unhealthy cholesterol in fried food and fatty food. All of those contain a lot of cholesterol in them. Now this reaches the liver in the form of a chylomicron from the intestines. It's just a carrier of cholesterol. One thing to keep in mind is when you eat more cholesterol, your body tries to balance that out by negative feedback. And so it tries to decrease the synthesis of cholesterol. It tries to decrease its absorption. And we excrete about 1 gram or 1,000 milligrams a day of cholesterol. And finally, we get cholesterol to the liver via transport from other places. So we have cholesterol some other place in the body, whether it's stored somewhere or that it's sitting in the arteries where we don't want it. So HDL, the good form of lipoprotein, is going to go pick up that cholesterol from everywhere else in the body and bring it back to the liver so we can use it, so we can metabolize, put it to use in some other way, because we don't want cholesterol just sitting in our arteries, for example. And this is called reverse cholesterol transport, and that's what makes HDLs the good type of lipoprotein, because they are bringing cholesterol from where we don't want it to be and putting it in the liver where we do want it to be. Now, those are the sources of cholesterol. Again, from the dietary cholesterol that we consume, we only absorb 50% of it. This is just to hammer in the point that the majority of the cholesterol that we have is not absorbed from the food that we eat. It's made by our bodies through de novo synthesis. And we will discuss the entire pathway of cholesterol synthesis coming up. But just remember that the majority of cholesterol we have in our bodies is through our own synthesis of cholesterol and not from food we eat. Now, let's just take an example. Let's assume you're consuming 400 milligrams of cholesterol in a day. We said 50% of it is absorbed, so you absorb 200. Then you have 200 that is unabsorbed, and then you have 800 that is being made from de novo synthesis, and that is how we're excreting 1 gram. So this is just, again, to hammer in the point that we're making most of our cholesterol and not consuming it. Now, if someone wants to lower their cholesterol, if it doesn't, after what I just explained, you might think that, okay, so it's not really about the diet anymore. It's about the making cholesterol part. So a very important point here is that in order to lower cholesterol, lowering cholesterol in the diet is not going to do much for you, because you're already not absorbing all of it. You're only absorbing half of that. So what you should really focus on is the making of cholesterol, why we're synthesizing so much of it. And so just keep in mind that lowering cholesterol in the diet is not going to have much effect on blood cholesterol, because most of it is being made by de novo synthesis. Okay, now before we get into the whole synthesis pathway and how we make it in the enzymes and everything, there are 2 enzymes that are involved in the transport of cholesterol around the body and the converting it from cholesterol to cholesterol ester. So I'll take this one by one. The first one is LCAT, or lecithin cholesterol acyl transferase, and this is the enzyme that converts it from a cholesterol to a cholesterol ester. Okay, so the enzyme which does that is LCAT, and from the name, acyl transferase, it's transferring an acyl group from lecithin to cholesterol. So you're taking a acyl group from a molecule called lecithin and transferring it to the molecule cholesterol, therefore leading to the formation of cholesterol ester. Okay, so here in the diagram, cholesterol to cholesterol ester via LCAT. All right. The second one is CETP, or cholesterol ester transfer protein, and this is from the name a transfer protein that is just taking a cholesterol ester from one lipoprotein and putting it on another lipoprotein. It's just helping with the transfer part of the process of moving cholesterol esters around the body. And so here you see a cholesterol ester inside an HDL, and CETP will take that cholesterol ester and put it on a VLDL remnant or a chylomicron, et cetera, in order to move it around from one lipoprotein to the other. Simple enough. So just remember 2 enzymes involved in transport and changing cholesterol to cholesterol esters. We have lecithin cholesterol acyl transferase, or LCAT, and CETP, or cholesterol ester transfer protein. Okay. Now let's get into the biosynthesis of cholesterol. So when it comes to biosynthesis that I've talked about and mentioned a million times by now, it's kind of similar to the ketogenic pathway, which making ketone bodies. I'm pretty sure that was mentioned in my good friend Rafa's and Rina's pals. If you haven't watched those, you should definitely watch them. But it's kind of similar to that in some enzymes, and you'll see when I discuss it later on. But one really important thing before we get into it is that to make cholesterol, our starting point is acetyl-CoA. All of the cholesterol we make, it will be made from acetyl-CoA. Acetyl-CoA is a molecule I'm sure you've heard of a million times by now, and that is just a 2-carbon molecule. And it comes from different sources, but it is the main starting point of cholesterol synthesis is acetyl-CoA. The second thing is that this process is highly regulated, and when I say highly regulated, it means that there is an enzyme involved. There's a rate limiting enzyme involved, and that's what makes it a highly regulated process. And the enzyme involved will be mentioned in the upcoming slide. Where does this biosynthesis happen? It happens in the liver, mostly in the liver. That's why I put a liver here to help it stick in your mind, but most of the cholesterol biosynthesis happens in the liver, but it also happens in intestines and as well as the skin and the adrenal cortex, the ovaries, the testes, but it's mostly in the liver. And the site is the liver cell cytoplasm. All right, so we're cooking cholesterol. We need some ingredients. What are the base ingredients? There are three elements that are most important in making cholesterol. Number one, as I mentioned, acetyl-CoA. Why do we need acetyl-CoA? Because that's our source of carbon atoms. We need carbon. If you remember earlier, what did I say? The cholesterol molecule is 27 carbons. So we need a source of carbon to give us all those carbons, and that comes from acetyl-CoA. Acetyl-CoA is a 2-carbon molecule, and it has multiple sources. The second thing we need is a reducing power, something to help reduce the molecules in the pathway of making cholesterol, and that is in the form of NADPH, which is just reduced NADP. Reduced meaning the hydrogen here. That's what makes it gives it reducing power, so that it can share that hydrogen to another molecule and reduce it. And lastly, ATP. So we need acetyl-CoA, NADPH for reducing power, and ATP for energy. The acetyl-CoA can either come from beta oxidation of fatty acids, for one, and that was mentioned in a previous lecture, or the dehydrogenation of pyruvate, or the oxidation of ketogenic amino acids. So if you can see here, all the acetyl-CoA is coming from fatty acid, pyruvate, amino acid. So this is the fat pathway, the carbohydrate pathway, the protein pathway. So when you have nutrients, they get broken down and form acetyl-CoA. You will then be able to use those ingredients to make cholesterol. Just keep that in mind. All right, let's get into the entire process of making cholesterol. And we'll divide this into 4 stages to make this easier. Now, these names look long and, a headache to memorize, but I'm sure you guys have had worse pathways in MOL by now. But the good thing is you don't need to know every single step of this pathway. The ones highlighted are the main ones you should remember, and I'll discuss exactly what you need to know in a minute. So let's start. We have 3 acetyl-CoAs. We take 3 of the acetyl-CoAs, and as mentioned, acetyl-CoA is a 2-carbon molecule. You take 3 of them, 2 times 3, you get a 6-carbon molecule, which is HMG-CoA, which I'm sure you guys have heard by now. HMG-CoA, or hydroxymethylglutarallyl-CoA, is the 6-carbon molecule as a result of adding 3 acetyl-CoAs together. This is the first step, okay? The second step, which is really important here, is that the conversion of HMG-CoA into mevalonic acid, and this is done by the action of the enzyme HMG-CoA reductase. This is, if you're going to take one thing from this lecture, is this step of this pathway. HMG-CoA reductase catalyzes the conversion of HMG-CoA into mevalonic acid. It reduces it. Reductase means reduces it. And how does it reduce it? As we mentioned, the reducing power of NADPH. This is the most important step in this pathway. It is the most vital step because HMG-CoA reductase is the rate-limiting enzyme, and that is where all the regulation will come from. So anything that wants to regulate the synthesis of cholesterol, it's going to act on HMG-CoA reductase. And any medication that we're going to discuss later on from diseases that happen because of cholesterol, it's going to act on HMG-CoA reductase. This is the most important thing in this entire lecture. This is the regulated rate-limiting step. The most important enzyme in this pathway is HMG-CoA reductase, which is reducing HMG-CoA into mevalonic acid. And that mevalonic acid will be converted into 5-carbon intermediates that are called isoprenoids. You don't need to know these long names, just know that it's being converted into isoprenoids. And then these isoprenoids will join to form squalene. Squalene is 30 carbons. So we're going from these 5-carbon intermediates, and we're adding them all together to end up with squalene, which is a 30-carbon molecule. Now, cholesterol is 27 carbons, so we need the last step of cyclizing that squalene and forming cholesterol, the 27-carbon molecule. So this is the pathway of de novo synthesis of cholesterol. Let's just go through it one more time. You start with acetyl-CoA, 3 molecules, you add them together, you get HMG-CoA, 6-carbon molecule. HMG-CoA is reduced by HMG-CoA reductase into mevalonic acid. Mevalonic acid is converted to 5-carbon intermediates that then join to form squalene, which is a 30-carbon molecule. Squalene is then cyclized to form cholesterol. And what did we need to make this? Acetyl-CoA, NADPH, and ATP. Okay, this is the pathway of cholesterol synthesis. And lastly, I cannot stress enough how important it is to just remember HMG-CoA reductase is the enzyme that is acting here. This is going to make up the majority of the questions about cholesterol. Now, we talked about the pathway. Let's talk about regulation. Again, the enzyme that is being regulated is HMG-CoA reductase. There are three mechanisms involved. Number one, covalent modification, and that is by phosphorylation and dephosphorylation. So what are the enzymes that are able to phosphorylate and dephosphorylate? We have kinases to phosphorylate the enzyme and phosphatases to dephosphorylate the enzyme. And which form is active and which one's inactive? This is very important to remember. The phosphorylated form is inactive, okay? The dephosphorylated form is active. So we're getting rid of the phosphate in order to activate the enzyme. Now, multiple things can affect these the phosphorylation, but two of which that I'm going to discuss is insulin and glucagon, and that's coming up next. But just remember, we phosphorylate HMG-CoA reductase in order to inactivate it, and that is by a kinase, while a phosphatase is going to dephosphorylate it and in which activate it and increase cholesterol synthesis. The second method of regulation is via gene expression. So the cholesterol levels, when they enter cells, they will be able to control transcription and translation and gene expression in order of making HMG-CoA reductase. And finally, degradation of the enzyme, just natural degradation. It has a half-life of 3 hours, and the half-life depends on the cholesterol levels. Now, covalent modification, the first regulatory mechanism that we mentioned. It is affected by insulin and glucagon. Let's break this down and make this, it's really simple and straightforward to understand if you link the feeding with the effective cholesterol synthesis. So you have insulin is released when? Insulin is released when you eat. So think of it this way. You're eating food, you have fats, you have proteins, you have carbohydrates. These fats, proteins, carbohydrates, they're being broken down into acetyl-CoA. They are providing you with a bunch of carbons. That's what you're eating. You have carbons in the food you eat. They're being broken down. You have sources of acetyl-CoA when you're eating. And now you have all this acetyl-CoA that you need to put to use. You don't want acetyl-CoA to go unused in your body. You want to make use of it. How do we make use of it? We make cholesterol out of it. So therefore, you eat, you have a bunch of carbons and acetyl-CoAs. You want to put it to use. Therefore, logically speaking, you want to increase cholesterol synthesis. You want to put that acetyl-CoA to use and start making the cholesterol. So you eat, what comes with eating? Insulin. This is how you tie it in altogether. It makes sense if you think of it just logically rather than just plainly memorize that insulin is going to increase cholesterol synthesis. So insulin will induce cholesterol synthesis. Now, how does it do this? Just like I explained in the previous slide, how do you activate the enzyme? You want to activate cholesterol synthesis, you want to activate HMG-CoA reductase. How do you do this? By dephosphorylating the enzyme. And by what do we dephosphorylate? Through phosphatase. Therefore, insulin will activate a protein phosphatase. Protein phosphatase will dephosphorylate HMG-CoA reductase, get rid of that phosphate group, therefore activating the enzyme HMG-CoA reductase. Once it's active, it will start making more and more and more cholesterol. So once again, feeding will promote cholesterol synthesis because when you eat, your body releases insulin, and insulin will then activate protein phosphatase. Phosphatase will dephosphorylate, therefore activating HMG-CoA reductase, leading to more cholesterol synthesis. Insulin also has two other ways of increasing cholesterol synthesis by providing more acetyl-CoAs because it's involved in glycolysis and making acetyl-CoA through that. And it provides NADPH from other pathways that it affects. Now, naturally, if insulin wants to increase cholesterol synthesis, obviously glucagon is going to do the opposite. Think of it logically once again. Glucagon is released when you what? When you fast, when you're not eating, when you're not taking in any food. You're not eating, you don't have sources of carbon, you don't have any acetyl-CoAs. Therefore, why would you try to make cholesterol if you don't have anything to make cholesterol from? And so therefore, glucagon is going to deactivate the enzyme. How do we deactivate the enzyme? By phosphorylating it. How do we phosphorylating it? By activating enzyme kinase. Okay? How does this? By glucagon will activate adenylyl cyclase. I'm sure you're familiar with this process. Activating adenylyl cyclase will lead to release of cyclic AMP. Cyclic AMP release will lead to activation of protein kinase A, and then protein kinase A will then phosphorylate HMG-CoA reductase, making it inactive, therefore making it not, therefore stopping cholesterol synthesis. Okay, so these two are having different effects on the pathway. And I just highlighted the part that is being activated, and glucagon is inactivating it. So I hope this is fully clear. It's very simple and logical if you think about it in terms of why it's happening the way it's happening. So fasting inhibits cholesterol synthesis. Feeding or eating will increase cholesterol synthesis. The second method of regulation is, as I mentioned, gene expression and transcriptional control. So cholesterol enters the cell, it will affect transcription of enzymes. This is the diagram. I just put it in words in order to understand what's going on. So cholesterol enters as cholesterol esters via LDLs, IDLs, and chylomicron remnants. Now it's in the cell, it gets converted to cholesterol. What does it do? Now think of it logically once again. You have cholesterol in the cell. You want to stop making more cholesterol. You want to start storing it. You want to, negative feedback. You want to keep things in balance. So when cholesterol increases inside the cell, it's going to have three goals. It wants to do three things. Number one, it wants to decrease the synthesis of cholesterol. You already have cholesterol. Why make more? So it wants to decrease the synthesis. The second thing it wants to do is it wants to increase the storage. So you want to start storing that cholesterol, put it in a storage place, and not just as free-form cholesterol. And finally, you want to decrease the uptake. So you've got a lot of it going in the cell. You want to close that door. You want to stop taking up all of that cholesterol through LDLs. Three goals. How does it do this? In three different ways. In order to decrease synthesis, of course, what do you think it's going to do? It's going to decrease HMG-CoA reductase. Simple as that. The enzyme that was the most important step in the pathway, it will stop the transcription and translation of it in order to decrease the level of the enzyme, therefore decreasing the synthesis. Straightforward enough. The second thing, when it wants to increase the storage of itself of cholesterol, it will increase an enzyme called acetyl coenzyme A acetyltransferase, new enzyme ACAT. And so when it increases this enzyme, it just increases the storage and putting away that cholesterol. The third thing it wants to do when it wants to decrease the uptake of cholesterol, it does it by decreasing LDL receptor synthesis. So here we have an LDL receptor that allows the LDL to bind and put in its cholesterol ester inside the cell. So when we want to stop this part, the uptake, we want to decrease the levels of LDL receptors on the cell surface. And so these are the three things that are affected by cholesterol inside the cell when it comes to transcriptional control and gene expression. So it will decrease HMG-CoA reductase to decrease synthesis of cholesterol. It will increase ACAT to increase storage. And finally, it will decrease LDL receptor synthesis in order to decrease the uptake of cholesterol itself. All right, so we mentioned, just to wrap things up a little bit, two regulatory mechanisms: covalent modification by insulin and glucagon, and gene expression in these three steps. And the third one, which was half-life, it has a half-life of 3 hours. That's all that there is to it, the half-life of HMG-CoA reductase. We're done with synthesis. We're done with regulation of synthesis. Let's talk, what do we do now with all this cholesterol? Cholesterol cannot be broken down in our body. We put it to use either in the functions that it has, or we get rid of it in some way, because it cannot be just naturally broken down back to acetyl-CoA. We can't reverse that process. So we have a few removal mechanisms. It can be lost in feces. It can be converted to bile salts. This is half of all cholesterol we have is converted to bile salts, and because bile is important, it has its own functions that will be mentioned. And finally, it can be reduced by bacteria to natural sterile molecules. Now let's get into the clinical stuff. We talked about all those things about cholesterol. We now correlate it with, real life, real world clinical things. So heart disease. I'm sure we all know of the, cholesterol gets its reputation because when you have very, very, very high cholesterol, that cholesterol builds up in the arteries. When it builds up in the arteries, it stiffens them and it tightens them and narrows the lumen. And when that happens, it can affect the blood flow through these arteries. And if it happens in the coronary arteries, because they're very narrow, that's why it affects it there. If this happens in the coronary arteries, it leads to coronary heart disease. And so this buildup is called atherosclerosis. And when it happens in the coronary arteries, it causes coronary heart disease. So increased levels of cholesterol is directly related to heart disease. And low levels of cholesterol, which is good, leads to less mortality, less morbidity, less atherosclerosis and heart disease, etc. All right. When you see this, it's like a real clinical condition that we're going to get deeper into. The first one is familial hypercholesterolemia. And this from its name. You have a lot of cholesterol in blood, and this is familial because it's an inherited mutation, genetics. So what is causing all of this cholesterol to be in the blood? It is caused by LDL receptor deficiency. You don't have an LDL receptor. Let me go back to the previous slide to explain this better. So if you don't have an LDL receptor, you cannot take up LDL cholesterol. So all of this LDL will be left in your blood. You don't want to leave all the LDL in your blood because LDL cholesterol is really bad for you. It's what causes atherosclerosis. It's what causes buildup of cholesterol and lipids in your arteries, and that leads to things such as heart disease. So you don't want to leave LDL in your blood. That is why, if the LDL receptor is not there, if there's a deficiency of LDL receptors, and you're leaving all the LDL in the blood, this can make a big problem, which is familial hypercholesterolemia. So this gene for the LDL receptor that is mutated is on chromosome 19. It's a recessive mutation, and depending on the severity of the disease, it can have the LDL receptor can have an activity of 0% of normal to 25% of normal, but regardless, it's just a lot less than you should have. And this hypercholesterolemia can lead to premature cases of coronary artery disease. So instead of getting coronary artery disease much later in life, it has a much earlier onset because of this condition. There are two forms. There's heterozygous, which is more common, and homozygous FH, which is much rarer. But the homozygous form is much more severe because you have extremely high levels of LDL cholesterol in the blood because you have a huge deficiency in LDL receptors. And therefore, this will lead to very early, or it's more likely to lead to very early symptomatic coronary artery disease. Now, how do we treat this? We could treat the heterozygous form, the less severe form, by, of course, changing the diet, eating more healthy food, trying to lose weight, trying to exercise more, just decrease the sources of LDL cholesterol to begin with. But other than lifestyle modifications, we can give the patient cholesterol-lowering drugs. And I'll be discussing that in the next slide. But when you have these drugs and you take these cholesterol-lowering drugs as well as lifestyle modifications, this can have, combined effect to upregulate LDL receptors to decrease LDL cholesterol. So what is this drug? The main drug that is used in the treatment of hypercholesterolemia is something called a statin. Statins are a group of drugs. For example, lovastatin, atorvastatin, they usually end in the statin, so they're easy to pick up. These were mentioned in your slides, but I doubt you need to memorize these names, Mevacor, Lipitor, whatever. Just remember lovastatin, statins, etc. These statins are structural analogs of HMG-CoA. HMG-CoA, if you remember, it was the second step of the pathway. And so it's a structural analog. It acts like it looks like HMG-CoA. So what it's going to do is it's going to go to the active site of HMG-CoA reductase and fit right into it and competitively inhibit the enzyme, therefore partially inhibiting the action of the enzyme, the rate-limiting enzyme HMG-CoA reductase. So this will lead to a 20 to 40% decrease in LDL cholesterol, and it will also help in increasing LDL receptors in the cell in order to get rid of all the LDL that is built up in the blood. It does have some pretty bad side effects like myopathy, but the main thing you need to understand here is how it's working and what it's doing, which is by inhibiting HMG-CoA reductase. This will be asked about. I've seen it be asked about in MOL exams before, because it's a very good representation of the function of HMG-CoA reductase because we're inhibiting it. Now, we're done with cholesterol. We talked about cholesterol esters, the function, the sources, the synthesis, the regulation, hypercholesterolemia, drugs. Now, once you're good with all that, let's move on with bile acids and bile salts. There are just a few definitions you need to understand the difference between these words: bile acid, a bile salt, and bile. Okay, and I want to clarify each of these things because you don't want to get confused between them because they're sort of different things. Bile acid, it's a steroid end product of cholesterol metabolism. So a bile acid is made from cholesterol. Okay, there are two forms: primary bile acids and secondary bile acids, which I'll explain in a minute. The second thing is a bile salt. A bile salt is also known as a conjugated bile acid. So it is when you take a bile acid and you conjugate it to glycine or taurine, amino acids, therefore making it a conjugated bile acid, which is the same thing as a bile salt. Okay? That is a bile acid and a bile salt. Now, what is bile? Bile is the liquid, the watery mixture that contains bile salts, but it also contains other things like phospholipids, bilirubin, etc. So bile is the liquid, okay? Now you know the difference between bile acid, bile salt, and bile. So just make sure you know the distinction, because when I mention each one, it's going to be a different thing. Bile is synthesized in the liver, but it's stored in the gallbladder. That's why I have a gallbladder picture here. And it's the major excretive form of cholesterol. So when we want to get rid of cholesterol, what we put it to use for, it's in order to make bile salts, to put them in bile, the liquid. And why we're doing this, the function of this is to, it's involved in dietary lipid digestion. So to digest lipids, because they act as an emulsifier. So it's really important in lipid digestion. Let's talk about primary and secondary bile acids. Primary bile acids are cholic acid and chenodeoxycholic acid. Okay, they're synthesized in the liver and they're made from cholesterol. How this happens is cholesterol is hydroxylated and then the side chain is cleaved, therefore making primary bile acid, hydroxylated by the CYP450 system. Okay, and after we make this bile acid from cholesterol, it can then be conjugated in order to make a bile salt, as just mentioned earlier, or it can be transformed into a secondary bile acid. Okay, and this primary bile acid is stored in the gallbladder. Secondary bile acids are deoxycholic acid and lithocholic acid. Okay, they are synthesized not in the liver but in the intestinal lumen, where there are a bunch of bacteria. This bacteria will take the primary bile acid and they will modify it in some ways in order to make a secondary bile acid. And the modification that it does is deconjugation and dehydroxylation. Okay, so primary bile acids made in the liver from cholesterol, secondary bile acids made in the intestinal lumen via intestinal bacterial modification from primary bile acids. Now, once again, okay, we talked about primary versus secondary bile acids. Let's touch base once again on the primary bile acids and conjugated bile salts. Okay, now bile acids, again, cholic acid, chenodeoxycholic acid, the OH group's on the same side. They have a pKa of 6, but I don't think it's important to remember at all. It's just the ionization number. It just means it's a weak acid, and they're good emulsifiers. However, the conjugated bile salts, which again are just bile acids that are conjugated to glycine or taurine, forming glycolate or taurocolate. These names sound weird, but think about it. Cholic acid also is known as cholate, okay? And when you add glycine to cholate, glycolate. If you add taurine to cholate, taurocolate. It's pretty straightforward if you think about it that way. Now, the bile salts are even better emulsifiers. That's why I make bile salts, because they're even better at emulsifying lipids. Great. This slide was not in your lecture. It's just to wrap all of those ideas up. We start with cholesterol. Cholesterol is then hydroxylated and we cleave the side chain to make a primary bile acid, cholic acid, and chenodeoxycholic acid. Then primary bile acids could be dehydroxylated in the intestinal lumen by the gut bacteria in order to make secondary bile acids, deoxycholic acid coming from cholic acid, lithocholic acid coming from chenodeoxycholic acid. And both primary and secondary bile acids could be conjugated in the liver with glycine or taurine to make bile salts or conjugated bile acids, such as glycolate and taurocolate. Okay, simple and straightforward enough. This is a summary slide of bile acids and bile salts. Now, why does this matter? Why are we talking about bile so much? Because there's something very common called gallstones or cholelithiasis. Theasis means stone, chole means, cholesterol, cholic acid, bile in general, bile salts, bile acids. They all have this part in their names. So gallstones happen when there is, you have a hypersaturation of cholesterol. When cholesterol gets hypersaturated, it starts to crystallize and form solid stones, and this is what leads to a gallstone. How does that hypersaturation happen? It's either when there's a deficiency of bile salt or when there's a hugely increased cholesterol excretion. So you have so much cholesterol trying to be excreted. Now, one way this can happen is when you're taking a drug like a cholesterol lowering drug like fibrates. These fibrates act to increase excretion of cholesterol. So you're trying to get rid of that cholesterol. And when you try to get rid of it, you're going to have a huge abundance of it going to the gallbladder, and that will lead to hypersaturation, you have so much cholesterol there, they will start to crystallize and form stones. Now, gallstones are usually asymptomatic. Some people are living their lives normally, having stones in their gallbladder, not even knowing that they have stones in their gallbladder, because it's if you think about it, if you just got stones sitting in your gallbladder, it's not really going to do much. It starts to be symptomatic when the stone starts moving in the bile duct and obstructs it, or the common bile duct, and this can lead to pain, inflammation. This is when problems happen, and this obstruction can lead to obstructive jaundice, for example, when the affects bilirubin metabolism. Now, how do we treat it when this happens? You just get rid of the whole thing. You surgically resect the entire gallbladder, and this is in a procedure called cholecystectomy. Chole, again, cholesterol, bile, all of that. Cyst is the gallbladder itself. Gallbladder is a cholecyst. It's the same name. Ectomy is a removal, cholecystectomy. Now, this is just a real life picture of what the gallbladder looks like. These are the cholesterol stones, the greenish hue on it. That's why we give bile that greenish color. Now, we talked about bile salts, bile acids, cholelithiasis, gallstones, and metabolism, what they all are. Talk about cholesterol, hypercholesterolemia, all the treatment of hypercholesterolemia. We're done with the cholesterol section. Before we move to the steroid hormone section, let's do a quick practice question. And this question is a very good example of everything that we learned and how these things can be asked to you. So, which of the following best explains why statin therapy is effective for individuals with hypercholesterolemia? So it's just asking what statins are doing in hypercholesterolemia. A, inhibiting HMG-CoA synthase, step 4, the synthesis of mevalonate that inhibits fatty acid synthesis. B, statins inhibit HMG-CoA reductase, a regulator of cholesterol synthesis. C, statins stimulate thiolase, thus making more malonyl-CoA for inhibition of the tricarboxylic acid cycle. D, statins bind to LDL receptors, displacing cholesterol, inhibiting cholesterol synthesis. Or E, statins stimulate the synthesis of trans-unsaturated fatty acids. You can, pause, take a look at the question, answer to yourself, do what you want, but the answer here should be pretty straightforward if you were listening. The answer is B, statins act by inhibiting HMG-CoA reductase, which is the key regulator of cholesterol synthesis. So this is a good example of everything we just discussed, a good example of how they can likely ask you, and keep in mind that they ask about statins a lot in terms of, not to test your pharma knowledge, but in order to test your understanding of the cholesterol synthesis pathway. Now, that's it for cholesterol synthesis and everything to do with cholesterol. Let's now move on to the second part of our lecture, which is to do with steroid hormones. Steroid hormones are just a group of steroids that function as hormones to modulate several physical processes. So again, they have a similar structure to cholesterol, but they're there to modulate processes. They act as hormones, pretty much. They have a few functions depending on the type of steroid hormone. We have glucocorticoids, such as cortisone and cortisol. These are involved in carbohydrate regulation. We have mineralocorticoids, which are involved in mineral balance. We have gonadal sex steroids that are involved in reproductive functions. They also have other functions like inflammatory response, stress response, bone metabolism, cardiovascular fitness, behavior, etc., etc., etc. But these are the main things you need to keep in mind. Now, before I talk about that slide, again, glucocorticoids are like cortisol, cortisone, mineralocorticoids like aldosterone, androgens like testosterone, estrogens like estradiol and estrone, and progestins like progesterone. I'm sure you guys are taking all about steroid hormones in Edari Pro, but this is part of the lecture, so I have to explain it. Now, the steroid hormones are, again, not water soluble. Everything we talked about in this lecture is pretty much not water soluble. But because they're not water soluble, they have to be carried in the blood complex to something specific called a binding globulin. And it's not like cholesterol where it's transformed into an ester and it's lipoproteins. No, steroid hormones are just, you bind it to a little binding globulin, which is soluble, in order to help it move around the blood. We have two examples to remember: the corticosteroid binding globulin, which carries cortisol, and the sex steroid binding globulin, which carries testosterone and estradiol, just as sex steroids. Okay, that is how steroid hormones are transported in the body, complex 2 binding globulins. All right, all the steroid hormones come from cholesterol, The only difference between them from mineralocorticoid to glucocorticoid is just the ring structure and the side chains. That's it, because they all are derived from the main molecule cholesterol. However, they're not packaged or stored. They're just synthesized and released into the body to have their effects wherever they need to, have their effect. And the enzymes which make them are in the mitochondria and smooth ER, and they are lipid soluble, not water soluble. And so if they're lipid soluble, that makes them easily able to move through cell membrane. So this will come later on in your other lectures. I'm sure it was mentioned in some physiology lecture somewhere. Steroid hormones, they don't need a transport protein. They don't need active transport channels or anything in order to move through the lipid bilayer. They just diffuse right through because they're very lipid soluble. So just keep that in mind. And they're not stored in cells. Now, cholesterol is the main precursor, but the one that comes after it that's a precursor to all of the steroid hormones is progestin, which is a type of progesterone. So just keep in mind, progestin is the biosynthetic precursor of all of the steroid hormones, and it comes from cholesterol. Okay, so I hope you can see this on my slide, but okay, now I can't read it. I just took this from the transcription, and the doctor said that questions regarding synthesis of steroid hormones will not be asked about in the context of MOL. So your doctor didn't go through this according to my knowledge, and they will not ask you about the process. The only thing you need to know is that cholesterol is a precursor, and you have multiple step reactions, and then you need to know the names and functions of the steroids. That's it. So I will not be explaining this. I'm sure you took it somewhere in endo report, probably, but I will not be going through the whole process. However, the enzymes involved, there's a lot of enzymes involved in the entire process. And again, the doctor said you don't need to study this whole table full of these enzymes. However, one that is the ones that are relevant are 21-hydroxylase and 11-beta-hydroxylase. The reason we need to know this is because their deficiencies of these enzymes are involved in congenital adrenal hyperplasia, which is a condition that I will be discussing, and it was mentioned in the lectures. So just remember that these two are implicated in the condition called congenital adrenal hyperplasia. Again, this is the whole pathway of steroid biosynthesis. The doctor said you don't need to know the details of the reaction, so I will not be going into it in detail, but I will talk about a few parts that are important to the condition we will be discussing in a second. And that is that we begin with cholesterol, and then cholesterol transforms into pregnenolone or progestins via the action of ACTH. ACTH, adrenocorticotropic hormone, is released from the pituitary in order to make the steroid hormones. And so this is important. Then at some point, you have the enzyme 21-alpha-hydroxylase, again mentioned right here. 21-alpha-hydroxylase is involved in the making of aldosterone and cortisol and corticosterone in both of these pathways. The next step in both of these pathways is catalyzed by 11-beta-hydroxylase, and 11-beta-hydroxylase is again implicated in congenital adrenal hyperplasia. So these are the two enzymes that I'm trying to emphasize mostly here, and that is in the process of making cortisol and aldosterone. Now, let's get into congenital adrenal hyperplasia. Let me try to move this up because it's kind of bothering. Okay, there we go. Congenital adrenal hyperplasia is a condition that is caused by the autosomal recessive defect in the enzymes that are responsible for production of cortisol. Now, okay, there are 3 subtypes of congenital adrenal hyperplasia, and these are characterized by the defect in each enzyme. So the first one, the most common one, is the deficiency in 21-alpha-hydroxylase. This makes up most cases of congenital adrenal hyperplasia. The second type of congenital adrenal hyperplasia is characterized by the deficiency of 11-beta-hydroxylase. Okay? The third type, which is very rare, I will not be discussing this, it wasn't mentioned in your lectures, is the absence of 17-alpha-hydroxylase. But this is just for your knowledge that there's a third type. So what is the pathophysiology here? Now, if you have a defect in these enzymes, just think of it like this. You have a defect of deficiency in either of these, you're not going to be able to make cortisol or aldosterone. And this lack of cortisol and aldosterone, your body senses it. Your pituitary gland senses it and goes like, okay, where's all the cortisol? Where's all the aldosterone? Why are we not making it? And therefore, your pituitary is going to start making even more and more and more ACTH. When you have all this ACTH, your ACTH goes to your adrenal gland and tells it, "Yalla, make all of the cortisol, make the aldosterone. What are you doing?" And so this ACTH is going to trigger the adrenal glands, and the adrenal glands are going to try to start making those hormones, but it can't because obviously it doesn't have those enzymes, so it's not able to make them successfully. But it tries, and when it tries to do that, it will end up undergoing hyperplasia and enlarging. So this is what happens. This is the pathophysiology. You have low cortisol, low aldosterone. That acts like a lack of negative feedback to the pituitary. Pituitary releases more ACTH in order to try to trigger the adrenals to make the hormones that it needs. Adrenals can't make them because they don't have the enzymes, but they're going to try, and that will lead to hyperplasia, enlargement of the glands, and as well as increased synthesis of the precursor steroids. So this is the general overview of congenital adrenal hyperplasia. All of this slide applies to all types, but there are differences between the types, and you need to know the differences. They're very important in order to tell between them. So let's start with 21 alpha hydroxylase deficiency. Now, this is written down for your, for you to connect the ideas written down in written form to memorize them. But let me explain here. So here's 21 alpha hydroxylase. If you have a deficiency in 21 alpha hydroxylase, you cannot make any of this stuff. Okay, you can't make aldosterone and you can't make cortisol. Again, the general thing which happens, more ACTH, more, hyperplasia of the adrenal gland. But what this leads to is you have low corticosterone, low cortisol, and low aldosterone. When you don't have aldosterone, you're going to have signs of mineralocorticoid deficiency. So the things that mineralocorticoids are supposed to do, they're not doing it. So what are mineralocorticoids supposed to do, which I'm sure you guys are aware of by now, but they're there to retain the salt and water. So this leads to when you have severe cases and there's high mineralocorticoid deficiency, you're not able to retain the salt and water in your body because you don't have aldosterone. And so this will lead to a loss of salt and water. You're going to lose a lot of salt and water, leading to hypovolemia, leading to things like shock. And when this happens in severe cases, when in a neonate, for example, an infant, this will lead to adrenal crisis, neonatal adrenal crisis. Now, there are less severe forms that they can present later in life, but this is just to illustrate the effects of not of having a dysfunctional pathway. Is that one thing that's going to happen is mineralocorticoid deficiency. Okay, now if you're not able to keep funneling them into this process, but you're releasing a lot of ACTH and you're still making pregnenolone, you're making all of these precursors, you need something to do with them. So they're all going to be funneled into this pathway, which is making androgens. And so you're going to end up with all of these going into this pathway rather than naturally making cortisol and aldosterone. And when you funnel them to this pathway, you end up with an excess of testosterone and androgens in general. And this will lead to signs of androgen excess. So you're going to have things like precocious puberty, hirsutism in females, just all signs of androgen excess can manifest in 21 alpha hydroxylase. So once again, you're going to have signs of mineralocorticoid deficiency, and you're going to have signs of androgen excess due to the lack of 21 alpha hydroxylase enzyme. You're unable to make cortisol and aldosterone. So all of that is summarized into this slide right here, illustrating the effects of the deficiency in 21 alpha hydroxylase. I hope that's clear enough because it's important. These clinical features are characteristic of 21 alpha hydroxylase deficiency. They're not like the other deficiency, which is 11 beta hydroxylase. This one is actually quite the opposite of 21 alpha hydroxylase deficiency. Again, it's written down for your use, but I want to explain this here just to illustrate it better. 11 beta hydroxylase deficiency, it's just the next step in the same pathway, but this will lead to wildly different effects. If you have a deficiency here, this will lead to this pathway being fully functional. Your 21 alpha hydroxylase is functioning. It's making 11-deoxycortisol and deoxycorticosterone. So these two precursors are mainly 11-deoxycorticosterone. When you have an excess of these, the thing about 11-deoxycorticosterone, or I'm just going to mention it as 11 or DOC. DOC, when you have an extreme overproduction of it because of the deficiency of 11 beta hydroxylase, when you have a lot of DOC, DOC acts like an aldosterone-like molecule. So it has weak aldosterone-like activity. So although you're not making aldosterone, you're not able to get the final product, which is aldosterone, you do have this precursor molecule called DOC, which has aldosterone-like activity. So when you have this deficiency and you have high levels of DOC, it can have the same effects as aldosterone. And these effects will manifest as mineralocorticoid excess. So the opposite of the previous deficiency. And mineralocorticoid excess manifests as the opposite again of what we just saw earlier, but it will lead to salt and water retention, volume expansion. So instead of hypovolemia, you have hypervolemia. And most importantly, and most characteristic of this deficiency is hypertension. So you're going to have hypertension because of the volume expansion, volume overload. You'll end up with hypertension. That's why I put this figure right here, just to like nail the idea in your head that the most characteristic thing in 11 beta hydroxylase deficiency form of CAH is hypertension. And that is not present in 21 hydroxylase deficiency. It's only present in 11 beta hydroxylase deficiency. All right, so just keep in mind, this will lead to mineralocorticoid excess, leading to hypertension. And again, the same thing that happened earlier will happen here, where you have excess of these precursors. They will funnel into testosterone, androgen pathway, leading to signs of androgen excess. And that will lead to the same things, which is precocious puberty, virilization, muscularization, things like that, just signs of androgen excess. So this slide summarizes in written form what I mentioned and explained here, but this should be pretty good to illustrate the idea in your head. Now, that finally wraps up everything to do with cholesterol, everything to do with steroid metabolism, the clinical conditions related to each one. I hope this was, made easily digestible and easy to understand for you guys and more clear. I tried to clarify most things that were in the lecture that weren't that clear from there. Now, please leave your feedback in this QR code if you'd like. If you have any questions at all, you can contact me on my number or on through my email. And I welcome any questions you guys may have. And yeah, I hope this was a beneficial session, inshallah. Don't hesitate to reach out. And yeah, I hope this benefited you guys and best of luck with your finals. Bitawfiq, inshallah.